Prostate cancer is the third most diagnosed cancer in Australia, with over 3,000 men dying every year. Prostate cancer occurs when abnormal cells in the prostate develop and continue to multiply uncontrollably. These cancerous cells can then travel from the primary tumour location to distant locations in the body, such as the bone. Avoidance of apoptosis is one of the major hallmarks of cancer cells. Apoptosis is regulated cell death, serving as a natural barrier to cancer development. However, tumours have created many strategies to limit or prevent apoptosis, ultimately resulting in tumour proliferation and survival.
Transforming growth factor beta, or TGF-beta, is a peptide that assists in regulating many cell processes, including cell proliferation, apoptosis, cell differentiation, and cell migration. Normally, TGF-beta inhibits proliferation and induces apoptosis in prostate epithelial cells, as well as early stages of prostate cancer cells. In early stages, TGF-beta is a ligand essential for TGF-beta receptor 2 induced apoptotic signaling through SMAD-dependent and independent pathways. The SMAD pathway involves TGF-beta ligand binding onto TGF-beta receptor 2, which recruits TGF-beta receptor 1, causing phosphorylation and activation of the receptor to then phosphorylate SMAD2 and SMAD3 proteins. These activated SMAD proteins then complex with SMAD4 and translocate into the nucleus to induce expression of apoptotic genes such as P15 and caspase enzymes. Prostate cancer cells have downregulated expression of TGF-beta receptor 2, reducing TGF-beta sensitivity. This downregulation is thought to be due to increased CpG methylation of the TGF-beta receptor 2 promoter by DNA methyltransferase, which prevents SP1 binding, which is a transcriptional activator. The loss and reduction of these receptors in prostate cancer leads to reduced apoptotic TGF-beta receptor 2 signaling, resulting in the evasion of cell death, malignant transformation, and increased cancer cell survival. The TGF-beta ligand is essential to stimulate this pathway. However, it has been shown that excessive levels promote angiogenesis and tumour progression via upregulation of VEGF. Therefore, regulation of the TGF-beta and TGF-beta receptor 2 expression levels are critical.
Our proposed drug will be an injection to the prostate which contains lentiviral plasmid that expresses customized TAL-TET1 fusion proteins, as described by Mader and his colleagues in 2013. The TAL repeat arrays can be programmed to specifically bind to TGF-beta receptor 2 promoter, and then the fused TET1 enzyme can specifically demethylate DNA, increasing TGF-beta receptor 2 expression. The injection will also contain nanoparticles with CRISPR-Cas9, inserting an aptazyme riboswitch gene upstream of the TGF-beta gene to control expression levels as described by studies by Zong and his colleagues. Riboswitches are a functional part of the mRNA transcript and can ultimately prevent it from being translated into a protein by inducing conformational change when the specific metabolite within the cellular environment is bound. By specifically designing an aptazyme riboswitch that can recognize the ligand TGF-beta, we can utilize this chemical control to regulate TGF-beta expression, causing it to be repressed when levels are high and upregulated should levels of growth factor drop. This allows critical regulation of TGF-beta expression, allowing appropriate levels to stimulate TGF-beta receptor 2 apoptosis, while also preventing overexpression and prostate tumour progression.
If we receive funding, the money would provide us with the resources to investigate the potential side effects and solutions for lentiviral transfection, which poses the risk of mutagenic insertion. Furthermore, as the transfection and TGF-beta receptor 2 promoter demethylation is not targeted specifically to tumour cells, there is a chance of upregulation of TGF-beta 2 receptors in normal cells. This can then increase the risk of apoptotic signaling and cell death of healthy cells. To date, artificial design and fully functional mammalian riboswitches have not been developed in the in vivo application. There have only been rational designs and engineering models as studied by Zong and colleagues. Therefore, funding is critical for future research and experimentation.
Overall, our novel drug proposal is a non-surgical treatment for prostate cancer that doesn't involve many of the risks associated with many of the current therapies, such as radiation therapy or surgery. Lentiviral transfection has proved successful in loose study on mouse models involving knockdown of metalloproteinase ADAM9 by intratumoral injection, supporting our therapeutic drug design delivery. It has the ability to be used as a long-term therapy as it uses mechanisms and pathways that are already in humans, which directly targets the tumour's ability to evade apoptosis, a major cancer hallmark.